class debate, and this is a topic that is very controversial among experts right now. And I'm very proud of my students in two groups prepared arguments to support opposing views. So the goal of this class activity is to develop critical thinking. So all the things that you learn in class, I'm sure you have a lot to talk about to each other in a civil manner, no fighting. So we have a schedule, so I'm not going to go through who's next. You know what to do next, and we'll just do it that way. Toward the end, there is a session for questions. So there will be questions from audience, and you will have a time to come up with the answer. So other than that, let's start the opening statement.
All right, so I want to start off by saying welcome, Steve guests and fellow peers. Today we set out to argue our point of view in circadian clock evolution. Now, when people think of circadian rhythms, some people might not know what that is, but basically what it is, it's your internal biological clock-keeping mechanism, and it regulates your daily functions in about a 24-hour period. So the importance of these circadian clocks is evident in the regulation of your metabolic functions, physiological changes, as well as your behavior patterns among all the domains of life. So scientists in recent years have been able to use genetic analysis in order to discover many of the mysteries regarding the molecular mechanisms and elements which actually make up these circadian timepieces. In the evolution of chronobiology, there have been two main schools of thought. There's been an independent evolution, which is the other team is arguing for, and our team is arguing for common circadian ancestry. Recent studies have identified and shown that there is evidence of a common circadian ancestor existing among the various organisms, such as the eukaryotes, prokaryotes, and even the archaea. So our group has found substantial evidence which supports our claim, and to demonstrate our point of view, we have discovered recent studies which support our argument. Currently, we have identified two main arguments. The first argument focuses on the peroxyredoxin, and the second argument focuses on the conservation of the molecular oscillator components. So as I said, the first argument is dealing with the peroxyredoxin proteins, and we're essentially saying that the oxidation reduction cycles are universal markers for all domains of life. The second argument focuses on the idea of conserved molecular components of feedback loops, which compose the various circadian oscillators. So the opposing view supports multiple independent clock origins within eukaryotes, and this seems rather unlikely when you look at all the evidence mounting, because there is notable evidence which suggests that there was a circadian clock in the archaea and the bacteria. This evidence reveals that natural selection has actually preferred circadian clocks even before the origin of eukaryotes. Additionally, the PRX oxidation rhythms resemble the rhythms of the cyanobacterial Cod-C phosphorylations. So this just shows that potentially there was an early metabolic clock in the common ancestor of both bacteria and eukaryotes. Overall, while the actual components may not necessarily be so highly conserved, the actual clock mechanism itself is conserved. An interesting thing to note is sequential rewiring may play a role in the substituting of one set of circuit elements with another non-homologous set, and this may be like a reason as to why you can see in between the plants and animals, they have non-homologous elements, but there's a common interconnected feedback loop in between them. So understanding the unifying factor of circadian clock among the different domains of life would open unlimited possibilities to target various metabolical, physiological, and even behavioral aspects of the cellular organism. Whether one believes in the origins of multiple origins or a common ancestry of circadian clocks, it's a really important question in the field of chronobiology that will no doubt open a door to new discoveries and understandings. Here we set out to settle this debate regarding the origin of circadian clocks and provide some insight on our perspective of common circadian ancestry. Thank you.
Hi, good afternoon, everyone. My name is Michael Phillips, and I am the leader of Team Multiple Origins. We would first like to thank Dr. Eric Klein and everyone for taking time out of their day to join us for what we hope is an educational debate about the origins of circadian rhythms. We would especially like to thank our professor, Dr. Kwon Lee, who shared with us his knowledge on circadian rhythms, and without him, none of this would be possible. We would now like to take the time to present Dr. Lee with a gift thanking him for everything.
Is this driving?
It's got your name.
Okay, so for those of you who
Time
don't,
is ticking.
for those of you who don't know, a circadian rhythm is a biological rhythm that persists under conditions of constant light, temperature, and environmental factors, with a period length of about 24 hours, whose phase can be reset by brief interruptions in the constant regimen, and whose period length is relatively independent of temperature within a physiological range of normal growth. These circadian rhythms have been identified in virtually all organisms from all kingdoms of life, ranging from bacteria to plants to animals. That being said, our team has researched throughout the semester about the origin and evolution of circadian rhythms. For our research, we believe that circadian rhythms have evolved independently from multiple ancestors and have differentiated further with evolution. Our arguments that support our view will show the differences in mechanism, production, and oscillation of rhythms amongst different species of multicellular and unicellular organisms, and how proteins differ from organism to organism suggests that there are independent origins of circadian rhythms. So we'd like everyone to sit back, relax, and enjoy the debate, and we hope that when you leave today, you go with the curiosity to search through some articles and try to find an understanding of the origin of circadian rhythms. Thank you.
So now we'll go for the first argument.
For our first argument, to support the idea that circadian clocks have developed from multiple origins, we believe that proteins differ from organism to organism and therefore suggest that they are from independent origins. The circadian proteins in cyanobacteria strongly support this argument. Cyanobacteria shows to have developed independently due to the fact that proteins that make up the circadian clock are very different from those of other organisms. Three essential genes that are particular in cyanobacteria are Chi-A, Chi-B, and Chi-C. In the paper of Baranaga M that was published in Science to find that these particular genes control the clock, mutations were made that affected the cyanobacteria's daily cycle. When only these three genes were restored, the 24-hour cycle also restored back to its normal function. More than two evolutionary origins are also possible. Cyanobacteria are thought to have risen from 2.4 billion years ago, and multicellular eukaryotes were not present until about 1.5 billion years ago. According to an article published by Rosbash M in 2009, this suggests that cyanobacteria rhythms were present way before eukaryotic rhythms existed. The different set of plant circadian proteins may have indicated a third independent origin. In Rosbach's article, he stated that there is no evidence that this relationship extended across the animal and bacterial kingdom. The key circadian proteins of mammals appear completely unrelated to the key circadian proteins of cyanobacteria. Therefore, given to the lack of evidence for common proteins and a common ancestor, it is unlikely that they have mechanistic relationship.
My name is Adil Gnadivo. And I'm Battle One. Although essential proteins within the circadian clock of cyanobacteria are different between other organisms, the endosymbiotic theory provides strong evidence that mitochondria and chloroplasts were once primitive photosynthetic cyanobacterial and proteobacterial cells. Based on what the endosymbiotic theory says, it states that a eukaryotic host cell ingested bacteria, so both became dependent on one another for survival, which resulted in a permanent relationship. This strongly suggests that the eukaryotic host and its endosymbiote have completed complex levels of integration, including its DNA, proteins, and molecular oscillations, forming a large spectrum of primary producers. Chloroplasts contain their own genome and DNA, so they cannot be made by a plant cell in which they must have been inherited. Their main function is to encode proteins essential for photosynthesis, which is where chlorophyll captures energy from the sun and stores it in ATP and ADPH while freeing water and oxygen. Photosynthesis is also known to be circadian regulated, in which the efficiency of it fluctuates with a diurnal circadian peak in the early to mid-light period. The same photosynthetic process was seen in cyanobacteria, so it could have been inherited by the eukaryotic host while it was an endosymbiote. Since this symbiosis was established, Ancestral cyanobacterial endosymbionts genomes have greatly been reduced with gene loss or transferred to the host nuclear genome. Most proteins found in modern chloroplasts are encoded in the nucleus, produced in the cytoplasm through plastid membranes, while the rest are synthesized into the chloroplast and then translocated. The expression nuclear encoded chloroplast genes are under circadian clock control. An example of this is nuclear encoded sigma factor 5. This controls circadian rhythms of transcription of several chloroplast genes and reveals one pathway by which the nuclear encoded circadian oscillator controls rhythms of chloroplast gene expression. Based on our research, we believe that photosynthesizing chloroplasts were free-living bacteria that had taken up residence inside the cells of many yeasts, protists, and chloroplasts. Inside the cells of many yeasts, protists, and even plants and animals. This suggests that clock mechanism of an ancestral cyanobacteria was spread through endosymbiosis into a host that later on evolved into eukaryotic plants. In conclusion, the endosymbiotic theory explains the relationships between cyanobacteria and other eukaryotic organisms and supports our claims that circadian oscillators originated from common ancestors, given that since cyanobacterial organisms sustained circadian oscillators were once living inside eukaryotic cells.
Okay.
So for our first argument, I would like to present the peroxyredoxin function as a common evolutionary mechanism preserved within the different species across all domains of life. We believe that there is a common ancestor to diverse circadian oscillators because there is a common metabolic mechanism, peroxyredoxin function, that is present in the species across different domains of life. Peroxyredoxin is a highly conserved family of antioxidant proteins that control intracellular peroxide level across all three phylogenic domains, archaea, bacteria, and eukarya. The important role of peroxyredoxin is elimination of the reactive oxygen species from metabolic byproduct. The function of peroxyredoxin is believed to be one of the original adaptational mechanisms between the organism to adjust to changes in atmospheric oxygen level in the beginning of anaerobic life. The first biological clock likely evolved 2.5 billion years ago, just after the Great Extinction event. And the environmental changes exposed organisms on the planet to harmful daily fluxes of reactive oxygen species and atmosphere. Peroxyredoxin protein cycle for oxidation and reduction is a 24-hour rate, which could have provided protection for organisms from cycling concentration of reactive oxygen species. Recent evidence shown that peroxyredoxin is a unifying factor demonstrating the preserved circadian clock mechanism between the different domains of life. There was a popular theory that the circadian rhyme originated only in DNA, but the recent studies show the evidence there is a possibility of this peroxyredoxin mechanism function. So one of the studies actually focused on the red blood cells, which is the most abundant source of peroxyredoxin proteins, and shows the evidence of the cycling rhymes. So for this study, the red blood cells were incubated in the dark at the body temperature and sampled in regular intervals for several days. And then they examined the biomedical markers, proteins called peroxyredoxin. And they observed that there was a 24-hour cycle, and that cycle actually was independent of DNA transcription and translation. It persisted even without that transcription translation. So for the different study, they observed in marine algae. They incubated those algae again in the dark and at constant temperature for several days, and observed again that rhyme persisted even in the absence of external cues. So even though research shown the circadian rhymes of peroxyredoxin persist in the absence of these cues, it also provided evidence that metabolic rhythm persists even in the second clock molecular transcription translation mentioned in planet's evolution. So even though the previous studies of evolution of chronobiology show some degree of individually evolved circadian clock between the different domains of life, the recent study disproved this point of view by example of peroxyredoxin.
According to the article, peroxyredoxins are considered markers for circadian rhythm by Rachel S. Eager and others. They they start by stating that the molecular phylogeny of mechanisms driving these rhythms has been difficult to dissect because identified clock genes and proteins are not conserved across the domains of life, bacteria, archaea, and eukaryotes. Understanding your claim that peroxidocins are highly conserved antioxidant proteins and control intracellular peroxide levels across the three domains, archaea, bacteria, and eukaryotes, I'd like to quote the article when they say, peroxidocin proteins constitute a universal marker for circadian rhythms in all domains of life by characterizing their oscillations in a variety of model organisms, end quote. So the peroxidocin may be universal among the organism, but all it is a marker. So what is a marker? A marker is just used to measure the timing of circadian rhythms. That's what the peroxidocins are used for. They are just markers, and the peroxidocin protein is not driving anything or interacting with the clock proteins. They are just present in all organisms, and they are just there to read out the metabolic activity. The article also went on to discuss how they tested whether rhythmic peroxidocin oxidation is required for timekeeping. The results obtained suggested peroxidocins are not even needed for proper functioning of an oscillator. I'd like to quote the article again when they say, this suggests that peroxidocins are not required for oscillator function in systems that possess an alternating timing mechanism, namely the transcription-translation feedback loop, end quote. So the peroxidocin markers were successful to observe rhythm in all domains of life in eukaryotes, but when they tested to represent the prokaryotes, they said that the system cannot represent a general prokaryote clock mechanism. They also went on to postulate that the peroxidocins represent circadian rhythms in cyanobacteria only in particular clock mechanisms they observed. However, this does not represent all the circadian clock mechanisms. For these reasons, we believe the peroxidocins are not evidence of common ancestry for a circadian rhythm perspective.
Just continuing off our rebuttal for the first argument, if you look at the screen, we have images of the clock oscillations for the different species. Up on the screen right now are the insects and the mammals. And although they all use proteins in order to conduct oscillations for their clocks, they all have different proteins, as you can see. For the insects, we have the PER10. For the mammals, we have the MPERs, MPER1 and 2 and 3. And then, John, if you go to the next slide. For fungi, they have the white collars, and then for the cyanobacteria, they have the chi, A, B, and C. And although all of them require proteins in order to conduct the oscillation, the fact that they all use different mechanisms to achieve the product, we believe that is an inclination of independent origin and multiple ancestry. So you're good for now. So moving on to our second argument, it focuses on the differences in the mechanism, production, and oscillation of the rhythms amongst different species. Multicellular organisms can partition clock function to generate different rhythms, while unicellular organisms require self-contained clocks that can generate a 24-hour rhythm. One may argue the fact that the presence of multiple oscillators in various organisms may denote the notion of clock mechanisms being derived from a common ancestry. However, the actual oscillations of the clocks work and regulate rhythms in significantly distinct manners, indicating independent originations. We will talk about the differences of clocks, oscillations between the two major groups of species, the unicellular and multicellular, and then later discuss the differences between the subgroups within each of the two major groups. In the unicellular organisms, mounting evidence indicates the existence of more than one oscillator within a single cell, each of which is predicted to consist of different components. For example, in the G. Polydera, two oscillators that respond to different wavelengths of light are thought to differentially regulate rhythms of bioluminescence and phototaxis. An example for the cyanobacteria involves the S. elongatus, in which a pacemaker that is based on the products of circadian genes clocks chi A, chi B, and chi C orchestrates a global rhythmic regulation of gene expression and controls the timing of cell division. However, the circadian timing mechanism itself is independent of the cell cycle. For fungi, studies show that cellular circadian clock of N. crassa is a network composed of coupled circadian oscillators, the pacemaker FWO, and one or more FLOs, and at least one other autonomous circadian oscillator, a second potential pacemaker that responds differently to environmental inputs and can direct diverse outputs. The presence of several oscillators probably contributes to the diverse rhythmic processes that are under clock control. for example, conjugation and the expressor genes that are unrelated of development. On the other hand, circadian complexity in multicellular species arises from the presence of molecular oscillators, which share the same molecular machinery in various cell types. The circadian system consists of light entrainable pacemaker in the brain that coordinates the rhythms of peripheral oscillators are however different. The suprachiasmatic nucleus has been shown to be a circadian pacemaker in mammals. Ablation of the mammalian SCN eliminates circadian patterns of behavioral activity, endocrine output, and many biochemical processes throughout the organism. The avian circadian system is even more complex than that of mammals, involving pacemakers that consist of two structures, the visual and the medial. However, the contribution of these pacemakers to the clock varies considerably even among the avian species. Another example supporting our argument involves the Drosophila melanogaster. The hierarchical model of the mammalian and avian circadian clock system does not hold for organisms such as the D. melanogaster, in which peripheral oscillators can be directly entrained by the cyclic environment cues. In the Drosophila melanogaster, the cell autonomous circadian timekeeping mechanism comprises two interlocked transcriptional feedback loops, and the clock genes that comprise these feedback loops are expressed in various tissues throughout the head, thorax, and the abdomen. And all our points that I just discussed, we came from the reference of the circadian rhythm multiple oscillators, of an article that we have presented to the opposing team.
Although various organisms produce varying circadian rhythms and regulate these rhythms differently, one cannot simply assume that this suggests that circadian oscillators did, in fact, evolve from multiple sources. It is true that multicellular organisms with differentiated tissues can partition clock function to generate and coordinate different rhythms, while unicellular organisms require standalone clocks, that can generate 24-hour rhythms for diverse processes. This is due to the fact, however, that multicellular organisms simply have more complex tissues that have their own molecular oscillators to provide rhythms for these different tissues. Unicellular organisms, on the other hand, make up for this deficit by having more complex oscillator systems in single cells because they are capable of having multiple oscillators in their single cell systems. So far, it is not known or discovered for multicellular organisms to have multiple clocks in a single cell. This information was also provided from the Circadian Rhythms of Multiple Oscillators Lessons from Diverse Organisms paper. While structural mechanisms do not necessarily seem to be preserved through the species and kingdoms, the core clock with at least one circadian oscillator is preserved. This core oscillator is common to all circadian clocks, which are universal and are found in cyanobacteria, fungi, plants, and animals. Even though the existence of this clock is universal, the clocks themselves may be used in different ways that best suit the organism. This suggests that the circadian clock may play an imperative role in allowing species to adapt to environmental niches that are in a way that is most biologically advantageous. According to Bay et al., it is, and I quote, "It is likely that the most significant evolutionarily conserved role of circadian clocks is to enable organisms to organize their physiology and behavior to occur at biologically advantageous times of the day." This is from the paper Circadian Regulation of Drosophila homologue and Mammalian Clock Gene, Per and Tim Function as Positive Regulators by Bay et al. Just because biological rhythms have different inputs and outputs does not mean that they arise from different sources. This is just another example of adapting to an environmental niche. For example, Drosophila melanogaster has a light-dependent circadian clock that has many similarities to the non-light-dependent mammalian clock. For mammals, the mouse was the specific biological organism representative. Homologous genes in these clocks suggest a high degree of conservation. Mammalian homologs for BMAL1, clock, and PER have been found and are homologous to the genes found in Drosophila melanogaster. Cryptochrome, or cry, a photoreceptor for the circadian clock of Drosophila, binds to the clock component TIM in a light-dependent fashion and blocks its function. In mammals, however, genetic evidence suggests that a role for cry is within the clock and is distinct from hypothetical photoreceptor functions. Cry, so this gene that is found in both mammals and Drosophila melanogaster in different forms, may have different functions in the two species, but it clearly comes from a common origin. Therefore, it is not unreasonable to assume that two clocks of different mechanistic natures can be from a common ancestor when they have many similar genes and functions.
Okay, so as Hiba mentioned earlier, our second argument is going to be based on the molecular oscillator conservation across the kingdoms. So we believe that there is a common ancestor for the diverse circadian oscillators because the basic molecular structure of the clock share the common interconnected feedback loops. We will discuss the commonality between the significant light givers, such as light and temperature, and the three main points of this discussion will be the conservation at the mechanistic level, the conservation of regulation, and the conservation of regulatory components. So first, we're going to discuss the light givers. So although the inputs may be different for these different organisms' feedback loops, the differences can be accounted for in different ecological niches, and that's just from natural evolution. The environment that they live in through natural selection changes the input for their feedback loop. And then we're going to show you some images just so you can see the commonalities. So this is the plant molecular oscillation. As you can see, CCA1 and LHY are positive regulators of PRR7 and PRR9, and PRR7 and PRR9 are negative regulators of CCA1 and LH1. And then for insects, DCLK and CYCL are positive regulators, and PER and TIN are negative regulators here. For fungus, white collar 1 and white collar 2 are positive regulators, and FREAK and FRH are negative regulators. And then for mammals, BMAL1, CLK are positive regulators, and PER2 and CRY are negative regulators. And then this is bacteria, which are obviously prokaryotes. Chi-A and Chi-B are positive regulators, and Chi-C is a negative regulator. And this is really important for our argument because, as most of you probably know, eukaryotes developed from prokaryotes. So the similarity in the way this loop works is very important because it means that the eukaryotic loop probably evolved from the prokaryotic loop. And then our last image, it just shows three, just to kind of bring home the similarity between them. Drosophila on the left is insect, Neurospora in the middle is fungi, and on the right is mouse or human, which is obviously mammal. And in this image, you can clearly see the similarity in the way that these loops work and the proteins and their function. And then the next thing we're going to talk about is the conservation of the regulation. So the core clock proteins are post-translationally regulated by phosphorylation, and the regulation of the RNA metabolism is also a possible commonality shown by recent research. And then the conservation of regulatory components means that the same kinases and phosphatases are involved in phosphorylation and dephosphorylation respectively, and these regulatory components are how we conserved.
First, I would like to begin by referring to Takao Kondo in his article, The Circadian Clock of Cyanobacteria. He begins by talking that the circadian clock of cyanobacteria have proteins encoded by clock genes in different organisms, and they do not share a sequence similarity. However, it appears more likely that the clock systems developed independently in cyanobacteria, fungi, and animals, and also possibly in plants. Natural selection by alternating environments apparently creates pressure to promote the evolution of similar clock proteins, but this idea should not be a surprise because negative feedback loop of gene expression is the rule in living cells rather than the exception. And for your claim about the interaction of plants, fungi, insects, and mammals interacting through the feedback loop, I'd like to refer to the article Peroxyredoxins are conserved markers of circadian rhythm by Rachel S. Eber. They quote by saying, while the clocks of eukaryotes like plants, fungi, and animals are largely understood in terms of transcription translation feedback loops, bacterial clocks are driven by protein-protein interactions over 24-hour cycles. They also continue to go on by saying that the transcription translation feedback loops components are not, however, shared between organisms. For example, the cyanobacterial clock is modeled around 3 proteins, Chi-A, B, and C. In the fungus, Neurospora, a loop involving the protein frequency and white collar complex is thought to drive cellular rhythms, whereas the plant transcription translation feedback loop involves elements including TOC1 and CCA1. Furthermore, although some multicellular organisms such as Drosophila and humans possess homologous components like the period proteins, their functions seem to differ between the organisms. Therefore, across all phylogenetic kingdoms, there are apparently no common clock components, suggesting that the daily timekeeping evolved independently within different lineages. For the point in regards to phosphorylation and dephosphorylation, it is true that the frequency and PER are phosphorylated by the same kinases and dephosphorylated by the same phosphates. But the frequency and PER proteins are not sequence homologs. What are sequence homologs? Sequence homologs are basically what it means that means that there is high sequence similarity. So what does this suggest? This suggests the high sequence similarity would occur because of convergent evolution, which is part of common ancestry. However, as previously said, frequency and PER are not sequence homologs, which would suggest circadian clocks have not evolved from a common ancestor, but an independent origin.
Are you done? Okay. Good job. So, if there's any question to our students, this is time. Can be any question. I have some, but I prefer you, yeah.
So in your research, have either groups found any examples of other strategies or mechanisms that are conserved across kingdoms but have very different bases, where the proteins involved are quite different, but the end result is the same process occurs? Have you come across any?
Okay, so what we're going to do is that we'll give you some time to think about it. So that's a good question. Any other question? Okay, I have a question for you. So you talked about, in response to the argument, you said endosymbiosis theory can resolve that issue. So according to your idea, you probably could find the protein similar to chi in chlorophyll or mitochondria, right? So I want you to answer whether you found that, or how would you respond. As far as I know, there's no homologous chi genes in chlorophyll. So I want you to answer that. And also their response. This is a following up your rebuttal that the PRX is this output, right? This sensing how not the rhythm generating mechanism. PRX is not part of the core molecular component of molecular oscillator, right? So that's just showing the rhythm. Yes, there's a rhythm. It reads the rhythm generated something we don't know yet, right? So it's not really arguing that there is a common oscillator, right? So think about that. So they rebuttal that, what about cry? So let's talk about insect clock and mammalian clock. You know that the cry in insect, like fly, is a photoreceptor, right? In butterfly, we talked about this, has two cry. One is involved in development, one is involved in photoreception. Mammal has a cry that is the core, not photoreceptor, but core of the. So it seems like when you look at those, okay, in insect, fly, and it seems like butterfly has two cry. One looks like insect cry, and one looks like mammalian cry. It seems like the middle of the transition from fly to mammal. So how would you explain that? As you said, independent evolution, then surely, you know, is there any connection between fly to mammalian clock? So with the Dr. Klein's question and my question, you have 2 to 3 minutes.
Well, you'll sprint upstairs and fail in a second, and then run back down. And you won't be out of breath at all, because you just smoked a minute. I'm actually trying to see it. I think we should time this. It's not evidence. No, I don't have to. Because that's all I'm on a cleanse. I have these beads behind my ears. Okay, I'm trying to be appropriate here. They're beads, and you release neurotransmitters from your brain telling you that you're full, and that increases your metabolism. And I've lost like 25 pounds in a month. So I
gained
mean,
25 pounds in 3, so.
well, so my plan is once I go off the diet, I'm gonna, maybe look it up on like NCBI, just validity.
No.
Look, I'm like decimated to just the eggs.
Yeah, I'm really bad at eating them. That is definitely true. You're not meant to be eating eggs.
I was like, wasn't the interest Dr. Lee and Dr.
I got you an email. I think I responded. I'm pretty sure. Yeah.
She just left, so.
Yeah, 99% sure I responded.
I think your movies from yogurt the new lab at the end of this week.
The movers are coming tomorrow. Yeah. No, I think that's the goal.
Yeah, but you still don't want to play with that,
Who needs an audit?
Oh, God.
Who needs like a $500,000 machine in the lab? Drop the
Common
bucket.
rail?
Doesn't it do it? Does it take 6 months? That's
No,
what
no,
it
no,
does.
we scrapped that. Small.
Okay.
Bigger than that tiny little one that we
Bigger
got,
than that
but
thing that
smaller.
we have one that can only fit a beaker.
So in Starbucks they teach us grande, not a tall, not a venti, a grande. Right?
It was literally this big.
Easy bake oven.
Okay, are you getting ready? Like 1 minute, think about the answer, wrap up.
Yeah.
We won't be able to plug it in now.
See it? Why not?
This is a 220 volt outlet. This is part of the renovations, I think. So we'll have it.
So if we put it in and close the door, and drop all drink, we still close it.
What about when you're racing?
Yeah, we're not doing that all here. So we're changing.
You're floating.
Yeah, so we're
Okay, so you want to go first? On answer, Circadian?
Sure. We're going to address the question about high A, B, or C's absence in chloroplasts or mitochondrial cells first. Here.
It's fine, I'll just talk loud. So we believe that it's possible that because of the chlorophyll and the chloroplasts, the photoreceptors that are in plants, that may be absent or not present in other animals like mammals or fungus or insects, etc., that could play a role in why plants did not develop a chi A, B, C like homologs the way other animals did.
Okay. Next question.
And then now we're going to address the peroxyredoxin question.
I'm just going to try to address this question because the study of peroxyredoxin is a very recent actually evidence of showing that there is additional metabolic mechanism of the function. But one of the study I found is actually they knocked out the mice gene producing peroxyredoxin, and the mice actually lifespan was significantly shorter, and mice explained anemia. That could be one of the possible evidence that the role of peroxyredoxin is very important. And in addition, those mice exhibited the behavioral rhythmic actually differences compared to normal mice due to circadian rhythm disruption in addition to anemia symptoms and lifespan shortening. So that one of the recent studies showing that there is more than just modulation in addition to transcriptional translational mechanism of DNA.
Answer to the Dr. Klein's question?
This is going to be a very honest answer. In our research, we didn't really come across any differences. However, I'm sure there are some out there, and it'd be wonderful to look them up and research them. But we didn't find any, so I'm sorry we cannot answer that question properly for you.
Thanks for being honest.
Thanks.
I respect that.
For Dr. Klein's question, we saw that photolyases are present in Arabidopsis, and they're the same member. They're a member of the same family, the Cryptochrome photolyase family, and they both do the same thing. They're DNA repair enzymes that use visible light to remove ultraviolet light-induced dimers. So the CPD-PP photolyases were found in Baturus, and they did the same thing.
And they're different proteins?
They were just part of the same family, but yet they were different proteins. They just did it a different way.
Can I add on to something?
Sure.
To that? Continuing to answer Dr. Klein's question, 2 days ago I actually had the opportunity to talk to another of our faculty member here, Dr. Jacoby, many of you know. I am in a genetics class, so I was just curious and asking him a few questions regarding these, and I asked him what his opinion was. He did point out a few things to me that kind of made sense to me and made my point on arguing for the multiple ancestors even stronger. He said the fact that if you look, John, if you could just pull up our PowerPoint up there, I can actually point out what he said to me. Just go, just go to the first 2 slides. If you look over here, for the insects, you have the per10 working as proteins to produce rhythms. In the mammals, you have the mPri, the mPro1 and 2, and the clock control genes producing the rhythms. If you go to the next slide, John, you will see the fungi and the cyanobacteria, the fungi working with the white collar genes, and the cyanobacteria working with the chi A, B, and Cs. All these proteins are different if you see amongst the different species. They're all doing the same job, producing rhythms. So that's why the fact that they are producing the same rhythm, yes, the end result is the same. The fact that they're using different mechanisms to produce these rhythms is what indicates evolution in our eyes. And that according to Dr. Jacoby, he says that is an inclination of independent evolution. I hope that answers your question. And then,
So one question down.
Could you reword your question? We were just a little confused. You said that the insects, within the insects, we have the flies and the butterflies that have the cry gene, but then with the butterflies, we have 2 cry genes. One
Look like insect, one look like mammal, like developmental.
Right.
And then mammal, the cry is not photoreceptor.
It's not, right.
So it seems like butterfly is in between fly and mammal. So what do you think about that? How would you answer it from your perspective?
So you're saying the fact that there is the presence of the cry gene in the butterfly that's solely for the purpose of development, you're saying they have similarity with the mammals, right?
Yes.
Why is that? In our opinion, why that is the case, you're saying?
So this is, apparently these cry are duplicated, right? This is very similar, but one has a function for photoreception, one has a function for development, right?
Right.
So mammal has only developmental cry. Fly has only photoreception cry.
Why does the butterfly?
Butterfly has both.
Right.
So are you saying fly, butterfly, mammal, they all evolved independently, have somehow butterfly come up with 2 cry, but mammal somehow have only 1 development? Does it make sense? That kind of points out that there's a continuum from insect to mammal. What do you think about that?
Well, a lot of the research that we found suggested that there is a common ancestor among insects and mammals that is different from bacteria because they do share similar proteins. I don't think it's up there right now, but.
So you're saying some eukaryotes have common ancestor?
Yeah, a lot of the papers that we found suggested that prokaryotes and eukaryotes have 2 independent origins for their clock mechanisms, and that the eukaryotes developed theirs independently of prokaryotes.
So you're kind of compromised. There is common
Right.
ancestor among eukaryotes. So
There may be common ancestors between some of the eukaryotes, but prokaryotes and eukaryotes themselves independently originated with different ancestors, what we're saying.
Okay, what do you think about this view? Do you think there is not really 1, but there is smaller number common ancestor in eukaryote, but prokaryote eukaryote are very different?
Well, I think the whole purpose of our research and study was trying to find that link. We didn't necessarily solidly come across this. That's why I think this question is really, it's open-ended. There's no right or wrong answer. And through our research, we kind of found that, yes, there's common ancestors seen among the eukaryotes, but that doesn't necessarily mean that holds over to the archaea or the bacteria. So that's the link that we didn't necessarily find. So like I said, it's just an open debate. In my eyes, there's no right or wrong answer. I do agree with some of their points, but I see where also we're coming from. So.
Okay, do you have anything that you want to say? If you don't say now, you cannot sleep tonight. Okay, so you have 2 minutes to make closing argument. You go first.
Alright, so for our closing, we hope that all of you learned quite a bit regarding the origins of circadian rhythms. We hope that we've somewhat convinced you that the differences in the mechanisms, production, and oscillation of rhythms amongst different species of multicellular and unicellular organisms, and how proteins differ from organism to organism, suggests that circadian rhythms evolve independently. We would once again like to thank everyone for coming out today, and again to thank Dr. Klein for being a guest judge and participating in our debate. This has been a very humbling experience for us. We realize that the study of circadian rhythm is an extremely vast subject, and that this debate contributed just a slight insight to the study of circadian rhythms. We would also hope that Dr. Lee, you enjoy your gift, and that years from now, when you look in the back of your closet, that you not only remember us, but also the entire class of Circadian Rhythm 2013. Thank you.
As you can see, there's a great deal of information supporting the idea that the circadian clock may have risen from a common ancestor. As mentioned before, peroxyredoxins, or PRX, demonstrates a common metabolic mechanism for circadian oscillators among many different species. Even in the absence of external cues such as light, PRX has continued to function. Aside from mammals, plants, and insects, research conducted has shown that the effects of PRX have been detected in both archaea and bacteria as well. Furthermore, circadian oscillators have been found to share common interconnected feedback loops. These intricate elements of the circadian clock have persisted across numerous kingdoms. Conservation of negative feedback loops, regulation of phosphorylation, and other regulatory components such as kinases and phosphatases further confirm that common ancestral argument. While it is true that there are stark differences between clock components in various orders of life, there are some substantial similarities among them which encourage individuals to think the previous notion of the independent evolution of clocks might need to be rethought.
Even though these conserved clock components may be employed in different ways by species, the different uses can potentially indicate the vital function that the circadian clocks have in allowing species to adapt to certain environmental niches. One thing to keep in mind is the common organization of the circadian clock among species suggests functional homology. Even though the debate regarding the origin of circadian clock is unresolved, this is an exciting time in the field of chronobiology, which we are constantly learning new information regarding the underlying mechanisms of the circadian clock. This new information has allowed scientists to reassess and further develop current clock models. Researching this question regarding the origin of circadian clocks will open a vast array of doors in the field of chronobiology, which will expand our understanding regarding the fundamental circadian clock mechanisms present in all organisms, regardless of what hypothesis you choose to support. We thank you all for your time and patience, and we'd like to take this time to thank Dr. Klein for assisting in the judging portion and Dr. Lee for coordinating this debate.
Okay, I think I'm very happy. You did a great job. Again, there's no right or wrong answer here, as you probably know by now. But the point was to actually doing your own research and reasoning and with open mind. So I'm very proud of you, and you did a good job. Okay? Thank you for coming. So let's take a picture.